which is a lightning talk. And our speaker is Alex Wade. He is a postdoctoral researcher at University College of London, focused on running simulations on high-performance computers. He's focused on simulations to investigate human proteins with a view to testing the efficacy of drugs and on designing new ones to treat diseases. The title of his talk is Optimizing Genomics Workflows for Neoverse. Alex, are you ready to proceed?
Yeah, I'm here. Thank you, Jeff. I'm going to be presenting in collaboration with my colleague from Atos. So Xavier, I invite you to introduce yourself as well, if you're there.
Yes, I'm here. Thank you, Alex. I'm Xavier Vigoureux. I'm based in France, as you can see here with my accent, and I'm taking care of collaborations, strategic collaborations. My focus is HPC, AI, and quantum. Thank you, Alex.
Yeah, sorry. So Xavier is going to be talking later about the work Atos will be doing on the new Neoverse platform. But for the introduction, I'll start by going through the application we're looking at. So this work is studying Mycobacterium tuberculosis, and this is the pathogenic bacteria which causes tuberculosis or TB in humans. So about 1.5 million people died from TB in 2020, and it's the 13th leading cause of death and the second leading infectious killer after COVID-19. In 2020, an estimated 10 million people fell ill with TB, and TB is present in all countries and all age groups. But the good news is that it's treatable and a preventable disease. So,
Alex, just to interrupt you real quick, are you sharing your screen?
I should be.
No, you don't, Alex.
It's okay. There's not too much relevant on the first slide.
Okay.
I think you can catch up.
Much better.
Okay, thank you.
Cheers. Yeah, so in 2020, an estimated 10 million people fell ill TB, but it's preventable. So the World Health Organization would like to eradicate TB by 2035, but to do this, we need better preventative and diagnostic methods. One of the main obstacles to this is the emergence of resistance, and this poses a major challenge to their objective, and it's the resistance that really this work would focus on specifically. So on this first slide, we have a visualization of some of the machinery that the bacteria uses to replicate itself, and zoomed in with the magnifying glass, we have a cartoon depiction of the drug situated inside of this machinery. So the machinery is just like proteins, and the different proteins perform different action in the human body as well as in the bacteria as well. The black hexagons here are depicting the drug, and around the outside is the protein chain, and each little guitar pick shape here is an amino acid, and it's the interaction of these amino acids with the drug which determine whether it's effective or not. If we zoom in further on the side chain of the protein, we'll see that this is just composed of atoms, which I represent on the right here as balls and sticks. And each group of atoms or block here is colored, and each one of these is an amino acid. You can write the amino acids just as a single letter. So in this case, you can then just write the whole chain as a series of letters, K, R, R, L, etc. Each of these amino acids is coded for in the genes of the bacteria, and we can get this sequence by taking a sample of the DNA of the bacteria and reading its DNA. The DNA is different between each sample of the bacteria as it evolves naturally and mutates, and we need to identify the relevant sequence of DNA out of the whole sequence of the bacteria's DNA, and accounting for the natural variation between different samples. And this is where sequence alignment comes in. So these are methods which allow you to search quickly through the sequence and match up the correlating parts of the DNA. once you have the sequence which is interested to you, which is of interest to you, you can start to look for individual differences in the amino acids between the mutant proteins. So here I show on the left the same sequence as before, and on the right I show a mutant protein. So the purple atoms which I've added are the atoms which are occurring in the mutant, and in the sequence at the top you can see that the S has changed to an L. So to work out whether this mutation confers resistance, we need to run a simulation of the normal protein and the mutant protein with and without the drug. So this essentially comes down to 4 simulations, and you're then comparing the interactions in the mutant protein and the interactions in the normal protein to determine whether the mutation has conferred resistance. So in all of this workflow, there's essentially 2 computational tasks, and this is the sequence alignment and the simulation. So for any predictions this method might make to be relevant to a clinical setting, that we need to get the results in hours. Days or weeks would be too long to be relevant to the patient. So it's really important that these pipelines are optimized to work as fast as possible. And I'll hand over to Xavier now to talk more about the work that Atos will do and maybe some of the optimizations that we will do with the ARM hardware. Xavier?
Yeah. So as you understood, there is a collaboration here between UCL, Oxford, ARM, and Atos, and this is really a joint effort that we are doing for the genomic. So first of all, I will try to introduce quickly what we are doing. Atos is providing supercomputers, so very high end, and in the top 500 list we are in. So really pure HPC. Obviously, for the numerical simulation now, there is also first large scale AI learning, which is interesting, and inference. And we consider that AI is also something that is modifying replacing some kernels in the numerical simulation that we had previously solved with usual simulation, usual numerical simulation. And the last one is the quantum, because we are currently preparing the new quantum era that will appear, that we hope will appear in the near future. So with what we are calling the QLM, it's a quantum learning machine. It's not quantum machine learning. So the goal is just to help the people interested in the quantum and to port their application on a simulator with up to 41 qubit, in fact, and to port their application on this machine to prepare the quantum computers for the NISQ era. So to come back to what we are doing in terms of collaborations with ARM and UCL and Oxford, you can go to the next slide. We are working on the platform. This is the platform, obviously the ARM Neoverse core, and the one we have chosen. In fact, this is not the V1 that we have chosen for this test, because we don't need what is brought by the V1 architecture with the SVE, for instance, with the large vectors and all this part of this floating point part. We consider that we will focus more on the bandwidth, and then this is why we have chosen the N1 architecture on which we will do the tests. So you know this one. And all the specification of this architecture. So today we just started to work on that. So this is really preliminary test we are doing on this machine. So I have no result to present. So we have this platform. We will have the application that is designed by Alex and by the UCL, and on the Atos side we are optimizing the usage of this platform. In terms of internal, if you go to the next slide, obviously also the Ampere Ampere, because this is a French mathematician. So the Ampere platform, so this is the one we will use. Again, the goal is to have this 80 cores running, N1 running for the binding for the genomic workflow. So we will have different stages. You have to understand that this, what's interesting in this collaboration is not to focus just on one part of the workflow, is really to start from the mutation that has explained that Alex just previously, and to see how the configuration is changed, and then having some genomics on higher level mutation recognition and understanding for the disease. So this is on this platform. This is, sorry for that, this is only preliminary results. So we have large memories, and we try to have all this workflow on a single platform provided by ARM. So I leave the floor back to Alex for this for the last two slides.
Yeah, thank you. Just to close out, I'll mention some of the larger simulations we're doing in this domain. So currently we have an implementation of the simulation side of this this workflow, and in general these are embarrassingly parallel simulations that we run, so we can scale these up to massive GPU counts, up to 26,000 on the bigger machines, and this allows us to get the results in the timescales which are required, which I mentioned previously is of the order of hours. And we'd be looking forward to deploying this kind of workflow on any future ARM-based exascale machines. And we're currently working on a cloud-based version of this, which might allow wider access to these kind of free energy, binding for energy calculations. And with that, I will end the presentation, and I'd like to thank all of the collaborators on this project. So everybody on the Atos team, Daniele and Phil, who I think I can see in the gallery listening, Peter Coveney and Zamin and Phil Fowler, who have given us a lot of the input models for these simulations, and David Whiffling at LRZ. Thank you.